Right, let's dive into some really cool stuff today. Stuff that's revolutionizing receptor research, next-gen tools and techniques. We've got scientific papers and even some YouTube lectures to unpack.
And it's cutting-edge stuff for sure.
It really is like going from blurry magnifying glasses to like high-definition 3D models.
Yeah.
And, scientists can even like tweak these tiny structures with incredible precision.
Yeah. Yeah, it's an exciting time to be in this field. A lot of breakthroughs are already leading to some really, really cool new therapeutic avenues for all kinds of diseases.
Okay, so one of the things that really stood out to me in all this research is just how much more complex receptors are than we used to think.
Mhm.
Like remember how we learned about signaling molecules like cyclic AMP just kind of flooding the cell, having a global effect?
Right.
Well, it turns out it's a lot more organized than that, a lot more localized.
It is, yeah. That older model is definitely being challenged these days. And there's this real world puzzle that really highlights this shift. So think about your heart muscle cells, cardiomyocytes.
Okay.
They've got these two different receptors. Beta-1 adrenergic receptors, those are activated by adrenaline, and then EP4 receptors, which are activated by prostaglandin E2. Now, both of these receptors, they trigger the same signaling cascade, the GS cascade, which eventually leads to the production of cyclic AMP.
So same pathway, same signal molecule. You'd think the effect on the cell would be the same.
Right. You would think so, right?
Yeah.
But here's the thing, it's not. Only stimulating the beta-1 adrenergic receptors actually increases the force of the heart muscle contractions.
Hmm.
Activating EP4 receptors, it does produce cyclic AMP, but it doesn't have that same effect on contractility.
So the same signal molecule can have different outcomes depending on which receptor it activates. That seems kind of counterintuitive.
It does, and it really shows the limitations of that older model we were talking about. So what's really cool is this emerging concept of microdomains or compartments, within the cell.
Okay.
It's like the cell has designated areas for specific cyclic AMP actions, almost like a cellular zip code system. You know what I mean?
So it's not just a free-for-all with cyclic AMP bouncing around randomly.
Not at all. Not at all. And this idea of localized signaling, it could explain why stimulating those different receptors on the cardiomyocyte, even though they both activate that same pathway, leads to such distinct effects.
Okay, but how does the cell create these microdomains? I mean, it just seems incredibly complex to keep everything organized like that.
It is. It's very complex. And that's where these proteins called akinase anchoring proteins, or AKSKPs, come in. They're essentially the architects of these signaling zones.
AKAPs. So they're like little traffic cops directing signals to the right place.
Yeah, that's a great way to put it, yeah. They act as scaffold proteins, localizing to very, very specific locations within the cell.
Mhm.
And here's the key. They bind to inactive protein kinase A, or pKa tetramers.
Oh,
Now, pKa, that's the enzyme that gets activated by cyclic AMP, and it's, super important for all kinds of cellular processes.
I see where this is going. So the AKA is holding the pKa in a specific location.
Yeah.
And when cyclic AMP is produced in that microdomain, boom, it activates the pKa that's already there.
Exactly. Yeah, it's a system of spatial control, making sure that the signal only gets activated in the right spot, leading to the specific effect that the cell needs at that moment.
Wow.
And get this, AKAPs actually bind to type 2 pKa over type 1 pKa, which adds another layer of specificity.
Wow, it's way more sophisticated than just a simple on-off switch, you know what I mean?
Yeah.
Like the cell is like a city, with different neighborhoods, each with its own like specialized services.
Yeah, that's a great analogy. And, this understanding of these microdomains is opening up all kinds of new avenues for research and potential therapies. Imagine being able to target drugs to specific microdomains, to like minimize side effects and maximize efficacy.
That's incredible. It makes you wonder what other levels of complexity we'll, uncover as we continue to kind of explore these signaling pathways.
Absolutely. And, we're really just getting started. Let's move on to another example that highlights this kind of multifaceted nature of receptors. Thyroid hormone receptors, or TRs.
From heart muscle to thyroid hormones. These receptors sure do get around. So what makes TRs so complex?
Well, there are two TR genes, TR alpha and TR beta, and each of them can produce multiple isoforms. These are like different versions of the same receptor, and each one has its own, specific function and expression pattern in different tissues.
So even though it's the same thyroid hormone, the effect it triggers can vary depending on which TR isoform is present in a particular cell.
Precisely. For example, some TR isoforms are really important for brain development, while others regulate metabolism or heart rate. It's another layer of complexity that shows just how, finely tuned our bodies really are.
Okay, I'm starting to get the picture here. Receptors are like multi-talented performers with different roles depending on, their context.
Yeah, and it gets even more interesting. TRs can actually act as both activators and silencers of gene expression.
Wait, how can they do both? I thought receptors were either, activators or inhibitors, not both.
It's a really cool mechanism. So in the absence of thyroid hormone, T3, TRs bind to DNA, and they actually repress gene expression.
So they're actively preventing certain genes from being expressed.
Exactly. But then when T3 comes along and binds to the TR, it's like flipping a switch. The TR transforms from a repressor into an activator, and it allows those genes to be expressed.
Wow, that's incredible. It's like a key unlocking a door to gene expression. But how does the TR know which genes to repress or activate?
There are several factors involved. First, the specific DNA sequence the TR binds to is super important. There are regions called negative thyroid hormone response elements, or NTREs, where TRs can actually repress genes even when T3 is present.
So even with the key there, the door might stay shut, depending on like the specific lock it's attached to.
Exactly. It's all about the specific interaction between the TR and the DNA. But it doesn't stop there. The TR's activity can also be modulated by interactions with other proteins, kind of like having a team of advisors influencing its decisions. You know what I mean?
Right, we saw that with AKAs and PKA.
Yeah.
Receptors are always part of a larger network.
Yeah, exactly. And in the case of TRs, they can even form partnerships with other nuclear receptors like RXR. These partnerships, we call them heterodimers. They can have, unique properties and regulate a whole different set of genes.
Okay, my brain is doing somersaults trying to keep up with all this complexity. So we've got different TR isoforms acting as both repressors and activators, depending on the DNA sequence, their interactions with other proteins, and even forming partnerships with other receptors.
You got it. It really is remarkable how much control is packed into these tiny molecules. But there's another layer to this whole intricate dance. TR function also relies on what we call a supporting cast of cofactors.
Cofactors. Are they like the stage crew for the whole gene expression show?
Huh. Yeah, that's a great way to put it. They are essential partners for both the silencing and the activating functions of TRs. We've got core pressors, which are involved in gene silencing, often by recruiting enzymes that kind of make the DNA less accessible.
Okay.
And then there are coactivators, which are involved in gene activation, and those do the opposite. They make the DNA more accessible.
So it's like a constant tug of war between these cofactors, with the TRs kind of acting as the referee, ultimately deciding which genes get expressed.
You got it. And all this just highlights how much we still don't know about receptors and all their intricate roles in cellular processes.
It's mind-boggling, and it makes me even more curious about, the tools and the techniques that are allowing us to kind of unravel these mysteries.
Well, that's a perfect segue. Let's move on to those cutting-edge tools. Welcome back to our deep dive.
Man, I was still trying to wrap my head around all that complexity we were talking about,
Yeah.
The microdomains, the TRs acting as both activators and repressors.
Right.
It's amazing how much control is packed into these tiny little molecules.
It really is, yeah. And as we delve deeper into this, the tools and techniques that we use are becoming even more sophisticated, and that gives us like a much clearer picture of how they actually work.
That's what I'm really excited to unpack, all those next-gen tools and techniques you mentioned.
Yeah.
It seems like we've entered like a whole new era of receptor exploration.
Yeah, absolutely. Yeah. One of the most revolutionary tools out there is cryo-electron microscopy, or cryo-EM for short. It's allowing us to see receptors at a level of detail that we never could before. It's like taking a snapshot frozen in time at like atomic resolution.
Cryo-EM. I've heard that term thrown around, but I'm not exactly sure what it entails. Do you break it down for me?
Yeah. So imagine being able to see the intricate details of a receptor's structure. Like its twists, turns, nooks, and crannies, all at the level of individual atoms. That's what cryo-EM does. It uses these beams of electrons to create these highly detailed 3D images of these molecules.
So it's like having a superpowered microscope that can see individual atoms like within a receptor. That's incredible.
It is?
But I always thought X-ray crystallography was kind of the gold standard for determining those molecular structures. So how does cryo-EM compare to that?
Yeah, that's a great question. So X-ray crystallography has been, the workhorse in this area for decades, but it has its limitations. it requires that molecule to be crystallized, which can be really, really difficult, especially for large, flexible molecules like receptors. It's
Yeah.
like trying to force a jellyfish into a perfectly square box.
I can see how that would be a problem.
Yeah.
So cryo-EM is kind of better suited for these more complex dynamic molecules because
Yeah.
it doesn't need that crystallization step, right?
Exactly. With cryo-EM, we can image receptors in their native state, so we're capturing their flexibility, their movement. It's like watching a movie of the receptor in action rather than just seeing like a single still photograph.
Yeah, that makes a lot of sense.
Yeah.
So we're getting like a much more realistic view of these molecules with cryo-EM.
Yeah.
So what kind of insights are we gaining from this level of detail?
Well, we can now see exactly how receptors interact with other molecules, like their signaling partners, drugs, even other receptors.
Hmm.
We can observe how those interactions actually change in response to different signals, and that gives us a much deeper understanding of how they function.
So we're not just seeing these like static structures. We're actually starting to understand the dynamics of how they work in real time, right?
Exactly, yeah. And this is a game changer for drug development.
Okay.
By understanding how a drug binds to a receptor at that atomic level, we can design much more targeted, more effective therapies.
That's really exciting. But, what about actually manipulating those receptors? Can we do that too?
Oh, absolutely. And that's where gene editing tools like CRISPR come in.
Ah, CRISPR, the revolutionary gene editing technology that seems to be everywhere these days.
Right.
How is it being used in receptor research?
So CRISPR is it's an incredibly powerful tool for dissecting the function of these receptors.
Yeah.
It allows us to make really specific changes to the genes that code for those receptors. So it's almost like having a molecular scalpel that can snip and stitch DNA with incredible precision.
So we can use CRISPR to create custom design receptors or delete specific parts of a receptor to see how that, affects its function.
Exactly, yeah. We can introduce mutations, delete entire genes, even insert new genes into cells. This lets us pinpoint the exact role of each receptor, its different domains, even specific amino acids within those domains.
It's like we're conducting these really controlled experiments, manipulating them at the genetics level to see what happens.
Yeah, that's a great way to think about it.
Wow.
And the things we're learning, from these experiments, they're helping us understand how those receptors contribute to diseases and then, develop new therapeutic strategies.
So we've got cryo-EM giving us this high-definition structural insights, and then CRISPR letting us edit the receptors with incredible precision. It's like we've gone from blurry photographs to being able to zoom in on the molecular blueprints, and we can even make edits to them.
Yeah, and it doesn't stop there. We also have these advanced biosensors that can actually track receptor signaling in real time.
Biosensors, that sounds like something out of a sci-fi movie.
Right. They might sound futuristic, but these biosensors are becoming really, really sophisticated. They allow us to monitor those dynamic interactions between the receptors and their signaling partners as they happen, right inside living cells.
Wow, so we can actually see those molecular conversations happening in real time.
We can. Yeah.
That's amazing. How do these biosensors work?
So a lot of them involve fluorescent proteins or other markers that are kind of attached to the receptor or its signaling partners.
Okay.
When that receptor gets activated, those markers change their fluorescence, and that gives us a readout of the signaling event.
So it's like having these tiny little spies inside the cell that are reporting back on what those receptors are actually doing.
Yeah, you could say that.
Wow.
And these real-time insights are so crucial for understanding those complexities of signaling networks and how they, contribute to different cellular processes.
It's like having a window into that secret world of the cell. We can see how quickly a receptor responds to a signal, how long it lasts, how it's relayed to other molecules.
Exactly. And this information is helping us, to develop much more precise and effective therapies, by targeting those signaling events that are critical for that particular disease.
This is mind-blowing stuff. So we've got cryo-EM giving us that high-resolution structural data, CRISPR allowing us to manipulate receptors with precision, and biosensors providing these real-time insights into those signaling events.
Yeah.
I mean, it's like a receptor researcher's dream come true.
It really is an exciting time, yeah. And all these tools and techniques, they're kind of converging to accelerate the development of those new therapies.
That's what it's all about, right? We want to use this knowledge to actually improve human health.
Absolutely. These advances are leading to therapies that are much more targeted, more effective, and have fewer side effects.
Mm.
They're opening up, this whole new world of possibilities for treating all kinds of diseases.
So can you give us some specific examples of how, like, these breakthroughs are being used to develop those new treatments?
Sure, yeah. Let's take GPCRs, G protein coupled receptors, for example. Those are the ones we were talking about earlier with the cardiomyocytes.
Right, right.
They're involved in tons of different physiological processes and are targeted by, many drugs that are currently available.
Yeah, I remember. But wasn't that process of, like, finding those drugs, like, kind of hit or miss?
It was, yeah. Traditional drug discovery often relied on just screening tons and tons of compounds to see if any of them happened to work. It was a very inefficient and time-consuming process.
So how are these new tools changing that?
Well, with those structural insights that we're getting from cryo-EM, we can actually design drugs that specifically target particular GPCRs now,
Hmm.
even specific conformations of those receptors.
Yeah.
It's like having a blueprint of the lock and then being able to design a key that fits perfectly.
So instead of just blindly throwing darts at a dartboard, we can now aim with precision.
Exactly. Yeah. And this structure-based drug design, it's leading to more potent and selective drugs that have fewer off-target effects, which means fewer side effects for patients.
That's fantastic. And what about CRISPR? How's that being used to develop these new treatments?
Well, CRISPR is being used to develop these cell-based therapies for all kinds of diseases.
Okay.
For example, in cancer immunotherapy, researchers are using CRISPR to actually engineer immune cells that can recognize and destroy cancer cells much more effectively.
So we're essentially like giving the immune system a boost, making it a more formidable opponent against the cancer?
Yeah, precisely. And these CRISPR engineered cells are showing, a lot of promise in clinical trials. They're offering new hope for patients who, haven't responded to those traditional
This
therapies.
is incredible. It seems like these next gen tools are like they're opening up this whole new world of possibilities for treating diseases.
They are. And the applications are just going to expand as we continue to, refine those tools and gain a deeper understanding of receptor biology.
This has been an incredible journey so far, but I'm kind of curious about the bigger picture here. We've talked a lot about individual receptors, their complexity, but how does this all fit into that overall network of cellular signaling?
That's a great question. Cells are, super complex systems. There are countless signaling pathways, and they're all interacting, influencing each other.
Right.
It's like this vast, intricate web.
So understanding those individual receptors, that's just the first step. We need to figure out how they're communicating with each other, how they're organized within those microdomains, and then how they contribute to like that overall flow of information within the cell.
Yeah, exactly. And that's where systems biology comes in. It's this field that really tries to understand those biological systems as a whole rather than focusing on just like those individual components.
So instead of looking at receptors, just in isolation, we need to think about how they're interacting with each other and with other signaling molecules to
Right.
create like this coordinated response.
Precisely. And this systems level understanding, it's essential for developing truly effective therapies that target not just individual receptors, but like those whole signaling networks.
Mhm.
It's like the difference between trying to fix one faulty wire in this complex machine versus understanding like the entire wiring diagram and how all the parts work together.
This is really making me think about the interconnectedness of everything within the cell.
Yeah.
It's like a really delicate ecosystem where even small changes can have these ripple effects throughout the whole system.
That's a great analogy. And it also highlights just how important it is to take like this multidisciplinary approach to receptor research where we're integrating all the insights from, structural biology, genetics, cell biology, and computational modeling.
So it sounds like a massive team effort with scientists from all these different fields coming together to kind of crack that code of cellular signaling.
Exactly. And that collaborative approach, it's really essential for pushing the boundaries of our knowledge and developing those innovative therapies that can like, address the root causes of those diseases.
This has been a truly like eye-opening deep dive into the world of receptors.
Yeah.
But I have to ask, welcome back. So before the break, you were talking about systems biology and how we need to understand how receptors work together, as part of these really, really intricate signaling networks.
Right.
It just seems like a monumental task.
It is a huge task, but advances in computational modeling and bioinformatics are really giving us powerful tools to tackle this challenge.
So we're talking about like computer simulations that can model these complex interactions, between receptors and signaling pathways.
Yeah, exactly. These models can incorporate like massive amounts of data from gene expression patterns, protein structures, to those real-time measurements of the signaling events we were talking about.
Right.
By simulating these different scenarios, we can start to understand the logic and the dynamics of these networks, almost like creating, a virtual cell.
That's a great way to put it, a virtual cell. And then we can run experiments in there, right?
Exactly.
And we can observe how those signals kind of propagate through the system.
Yeah.
We can tweak things, see what happens when a receptor malfunctions, maybe even test the effects of different drugs.
Exactly. And then that lets us make predictions about how the system might respond to different perturbations, like drugs, genetic mutations, which is, incredibly valuable for drug discovery, for personalized medicine.
So instead of just studying individual receptors, in isolation,
Hmm.
we can now start to see how they work within the context of the whole cell, maybe even beyond.
Yeah, that's right. Systems biology is really allowing us to connect the dots from, that molecular level all the way up to tissues and organs and helping us understand how those complex interactions contribute to health and disease.
This really highlights, that interconnectedness of everything in
Yeah,
biology, doesn't
yeah,
it?
it
It's
does.
like a giant puzzle, where each piece, from the genes to the proteins, the cells, it all kind of contributes to that bigger picture.
It does. And understanding that bigger picture is really essential for developing effective therapies that target the root causes of disease rather than
Uh,
just, treating the symptoms. It's
Right.
like the difference between patching a leak in a pipe and, understanding the entire plumbing system of a house.
So with all these advancements in receptor research, what does the future hold? Where do we go from here?
I think we're entering like a golden age of discovery in this area. We've got these powerful tools, the insights from systems biology and computational modeling. I mean, we're poised to make some truly transformative breakthroughs.
Can you give us like a little glimpse into what some of those breakthroughs might look like?
Yeah, I think we'll see personalized therapies, that are tailored to a person's unique genetic and molecular profile. Imagine a world where treatments are designed based on the specific receptors and pathways that are, messed up in a patient's disease.
That sounds like science fiction.
Yeah.
But with all these advances, it's becoming more and more realistic.
It really is. And I also think we'll see new therapeutic modalities, things like gene therapy and cell-based therapies that directly target those receptors and their pathways. I mean, we're seeing it already with those CRISPR-engineered immune cells for cancer immunotherapy.
It's really exciting. It feels like we're on the cusp of like a revolution in medicine
I agree.
where we can finally address diseases at their most fundamental level.
Yeah. And it's all thanks to the amazing researchers all around the world who are, just driven by curiosity and a desire to like unravel the mysteries of life.
Well, this has been an incredible deep dive into the world of receptors. And I feel like I've learned so much. Any final thoughts before we wrap things up?
I think the most important thing to remember is that we're really just scratching the surface,
Yeah.
We're only beginning to understand how complex and how important these receptors are.
Right.
They play such a crucial role in like everything, every aspect of our biology.
Mhm.
And, the more we learn about them, the closer we get to unlocking those new treatments for all those diseases we've been talking about.
That's a perfect note to end on. Thanks for joining us on this incredible deep dive into the world of receptors. It's been a fascinating journey, and I can't wait to see what the future holds, for this exciting field of research.